Our second mechanism of evolution is genetic drift. Genetic drift involves random changes in allele frequencies of a population. This means that sometimes we see allele frequencies changing just due to random chance, like the roll of the dice. In the example shown here, cleft chin is a heritable trait, and it is dominant. The allele frequencies in human populations will drift or change randomly up and down from year to year. This is because neither one of these alleles is related to reproductive success. Inheritance is based solely on chance. We can see that the impacts of genetic drift are much greater in small populations than they are in large populations. In small populations, it is easy to lose alleles, and this can lead us to fixation when we see no variability left in the population. Fixation is when there's only one allele, and that allele has a frequency of 100%. Often in populations, we see that fixation occurs with negative traits, things like diseases or problematic genes. Genetic drift in general tends to reduce genetic variations in a population, which is a negative effect and can cause a great deal of issues and problems. There are two effects that can cause genetic drift. The first one is the founder effect. A small number of individuals leave a larger population and become the founding members of a new isolated population. This founding population will have different allele frequencies than the original population. The individuals who have left the original population have been randomly selected. One example of a founder effect is in the Amish population. If we look at the original population in Europe, there is a very low occurrence of the allele that codes for polydactyly, or extra digits on the hands and toes. When the Amish left England, it was a random group, random in respect to their genes, that came to settle in the colonies, now the United States. However, a large proportion of those new founders had the allele for polydactyly. We now see that this population of Amish in these states has a high occurrence of the gene and of the trait polydactyly, much higher than the original population. Another effect that can cause genetic drift is the population bottleneck. Bottleneck effect is when a large proportion of a population is killed off due to natural disaster. The individuals that die randomly, not based on their genes or any particular trait. After a bottleneck effect, the surviving population will have a very different allele frequency than the original population. A bottleneck effect has been seen in cheetah populations in Africa. Originally, the population had a great deal of genetic variation. After the bottleneck event, which was caused by human interactions, destruction of habitat, and humans hunting them, we see that the population now has very few alleles and that the variation is very low compared to that original population.